Hello, my name is Shilpa Dange, and I work with Dr. Assefa and Dr. Koehler on my Archaea in mammalian gut microbiomes. Humans are superorganisms consisting of about 30 trillion human cells, and microbial cells make up to 39 trillion cells. So we have more microbial cells than human cells. Every human has 23,000 genes, and human microbiome has 2 to 20 million genes. So the question is, what is a microbiome? Microbiome includes microbiota, which includes bacteria, archaea, fungi, and viruses, and their microbial genomes. So these microbiomes, along with their genomes, are present inside our bodies and on our bodies at different locations. I will be focusing on gut microbiome. Archaea is one of the most enigmatic domains of the three domains of life, the other two being bacteria and eukaryota. So archaea are unique in that they share similarities with bacteria, such as that they are prokaryotes, have 78 ribosomes like bacteria. Archaea share characteristics with eukaryota in that they have transcription, translation, and some metabolic pathways similar to eukaryota. Archaea are unique in themselves in that they have cell walls with pseudopeptidoglycan, which causes or makes the archaea resistant to lysozyme and other antibiotics. Methods and materials first include sample collection from small intestine and colon. These samples are called as digesta, and pellets are collected from feces. DNA is extracted from digesta as well as pellets. After DNA is extracted, two different sequencing techniques are used. The first technique is called as 16S rRNA gene sequencing that tells you which archaeal and bacterial 16S rRNA gene sequences are present in your samples. Metagenomic sequences or sequencing tells you what genomic sequences derived from archaea and bacteria are in the sample. 16S rRNA gene, this is what it looks like, and this gene has 9 hypervariable regions, and the most commonly sequenced variable regions are V3 and V4. So the two disadvantages about this technique is that this technique can only target or identify and differentiate bacteria and archaea only, and this technique allows for taxonomic profiling to genus level only. It is relatively cheap. Whole genome shotgun metagenomics is expensive, allows for assembly of microbial genomes. This technique allows for taxonomic profiling to strain level, which is an advantage. The disadvantage is that the data analysis is more challenging. Now, how does this work? Here we have genomes of microorganisms found in the samples. DNA is extracted. Genomes are fragmented randomly. Small DNA fragments are sequenced. A small sequence reads are assembled into context. Genomes can be assembled using reference database or de novo assembly for unknown genome. So role of archaea in health and disease is unknown. Many believe that archaea are non-pathogenic and don't cause disease. Others believe that archaea are pathogenic and cause disease. A recent proposal on an archaeal strain called as Methanomesylicoccus luminiensis was proposed as a potential archaebiotics. Archaebiotics are similar to probiotics, but probiotics uses bacteria. Archaebiotics will be using archaea. These are the metagenomic data at different taxonomic levels. Figure 1 shows relative abundance of bacteria and archaea at kingdom level from 6 small intestinal samples of rats. Blue color indicates bacteria, green color indicates archaea. In fact, sample 1, as you can tell from sample 1, there is very little to no archaea present. So archaea are generally present in lower abundance as compared to bacteria. Figure 2 shows relative abundance of bacteria and archaea at the family level. Different families are color-coded differently. Green color indicates families from archaea. One such family is Arnatri albaceae. Figure 4 shows a Sunburst chart that shows taxonomy from kingdom to strain level. Blue color indicates bacteria, orange color represents archaea, and archaea make up only about 2% of the entire microbiome. So figure 5 is that 2% elaborated and shows Archaea and diversity of Archaea. Conclusion: studies indicate that Archaea can be found in lower abundance in small intestine and colon. Both 16S and metagenomic studies show presence of low quantities of Archaea in samples. However, whole genome shotgun metagenomics are more reliable to detect Archaea. Future studies include confirmation of Archaea using qPCR. Hopefully, we will have some answers to the role of Archaea in microbial community and in health and disease. Further research will include analysis of human samples. Here are my references. Thank you.